Imagine a world where drones and advanced 3D models are revolutionizing our understanding of wheat genetics, leading to more resilient and productive crops. This isn't just a futuristic dream. It's a reality that researchers are making happen today. In a groundbreaking study published in Plant Phenomics on February 27, 2025, a team led by Yuntao Ma and Yonggui Xiao from China Agricultural University and the Chinese Academy of Agricultural Sciences has demonstrated how low-cost drones equipped with specialized imaging technology can provide unprecedented insights into wheat plant height. By capturing multiple height measurements instead of relying on a single average, this innovative approach reveals subtle variations within crop plots, leading to more accurate genetic mapping. Wheat, scientifically known as Triticum aestivum L, is a staple food that contributes nearly 20% of the world's caloric intake. One of the most critical traits in wheat cultivation is plant height, pH, which affects both yield potential and the plant's ability to resist lodging or falling over due to heavy winds or rain. Historically, during the Green Revolution, the introduction of dwarfing genes significantly increased wheat yields globally. However, modern breeding programs continue to seek the optimal plant height to balance productivity and resilience. Traditional methods of measuring plant height involve manually gauging a few representative plants in a field. These methods are not only labor-intensive and prone to human error, but also fail to capture the full range of variation within a plot. Enter high-throughput phenotyping, particularly 3D canopy modeling, which offers a more precise and unbiased way to assess plant height. The study utilized low-altitude unmanned aerial vehicle (UAV) cross-circling oblique CCO imaging to measure wheat plant height across various environments. This technique was compared with conventional nadir imaging, where both methods were tested at the same flight altitude and overlap settings. The CCO imaging captured more detailed canopy structures, especially at the front of the plots, producing denser and more accurate point clouds. By extracting height data from 11 different quantiles, the researchers found that CCO imaging had consistently higher correlation coefficients and lower root mean square errors, RMSEs, compared to nadir imaging. The 90th percentile of the height quantiles most closely matched field-measured plant height, FMPH, while lower quantiles risked measuring stem height instead of the canopy. The detailed point clouds generated by CCO imaging allowed for the reconstruction of canopy structures at the organ level, clearly showing spikes, although side views were limited in closely spaced plots. Analysis of recombinant inbred line RIL populations showed that both FMPH and multi-level 3D pH followed normal distributions, with strong correlations across quantiles and high broad-sense heritability, ranging from 0.775 to 0.959 within environments, and 0.975 to 0.982 across environments. The 90th and 92nd percentiles achieved RMSEs below 2 centimeters in most cases, with a maximum correlation of 0.99 between FMPH and 3D pH. Quantitative trait locus, QTL mapping, across 7 environments identified 106 loci for FMPH and 3D pH, with 40 shared loci and 11 stable loci unique to multi-level 3D pH. 2 potential novel loci, QPH-ZJ.Cas.3a and QPH-ZJ.7a.1, were converted into CASP markers, validated in natural populations, and linked to significant plant height differences under varying irrigation conditions. Candidate gene analysis highlighted RHT5, a gibberellin-sensitive dwarfing gene on chromosome 3. Both genes were supported by CASP validation, further confirming their importance in plant height regulation. But here's where it gets controversial. While the integration of UAV CCO imaging and multi-level 3D pH analysis offers a powerful tool for breeders, some critics argue that the technology might be too costly for small-scale farmers in developing countries. However, the low-cost nature of the CCO imaging system and its scalability make it a promising solution for advancing precision agriculture and crop improvement. This workflow, which combines UAV CCO imaging and multi-level 3D pH analysis, provides a low-cost, scalable, and precise tool for phenotyping plant height. By uncovering genetic loci that traditional methods might miss, this approach enhances the efficiency of marker-assisted selection, accelerating the development of high-yield, lodging-resistant wheat varieties. Beyond wheat, this methodology could be adapted for other crops where canopy architecture and height are crucial, opening up new possibilities in precision agriculture. What do you think? Is the cost of implementing such advanced technology worth the benefits in crop improvement, or should we focus on simpler, more accessible solutions? Share your thoughts in the comments below.